Hi, everyone. Thank you very much for coming to my talk. Today, I will share with you my research on microplastic contamination in an urban bay, San Francisco Bay, and what we learned about its novel sources and fate. It is estimated that 4 to 12 million metric tons of plastic enter the oceans annually. There are a variety of sources of plastic pollution that exist, both primary and secondary, with more than 80% of them being terrestrial. Microplastics make up for the majority of plastic pollution in the environment, and by definition, they are less than 5 millimeters in diameter. Why we're so interested in microplastics is because they have a greater potential to interact with living organisms and to infiltrate food webs. A better understanding of regional sources and fate of microplastics can help to inform policy and to mitigate their inputs. For this study, we investigated microplastic contamination in San Francisco Bay, an urban bay in California. The bay is divided into 4 main regions. The north region consists of San Pablo and Suisun Bay, and then we have the central portion, South and Lower South Bays. We're interested in investigating the bay because there are a variety of sources that could contribute microplastic to this region. That's why we're interested in studying it. And also, we took samples from the adjacent national marine sanctuaries in ocean water beside the bay to investigate the microplastic contamination levels there as well. The experimental design of this study involves sampling a variety of different samples from the bay. And then once we've extracted and quantified the debris within these samples, we compare their communities to determine what sources are important and what we can do to mitigate these inputs. So the 6 different sample types that we collected are 1 liter crab, manta, trawl, sediment, fish, stormwater, and wastewater. Due to our uneven sampling design, we're unable to conduct statistical analyses, but we used a variety of qualitative statistical analysis methods to explore our data. In the lab, as we were extracting our plastic particles, we looked for 7 distinct types of morphologies, which are listed here. And as you can see, our samples are very contaminated. Our overall results are, starting with concentration trends, on average, we found that the highest concentration of microplastics tend to be in Central Bay. This is true for 1 liter and manta trawl samples, as well as our 2 source water samples, stormwater and wastewater. We think this is the case because a large portion of the population resides in that area, so they may be contributing plastic to surface water that way. We also found huge amounts of microplastic in stormwater compared to wastewater. On average, stormwater contained roughly 140 times more microplastic than wastewater. So this difference is huge. And notably, we found a lot of black rubbery fragments, which we suspect to be tire dust in sediment. And we found that sediment contained, on average, the highest microplastic concentrations in the lower south region. And this could be due to limited flushing, resulting in the microplastics being trapped in that region. We also found that depending on seasonality, depth of the bay where we sampled, and surface currents, the concentrations of microplastics were influenced by these environmental variables, so they differed. In terms of morphology and material type trends, overall we found huge amounts of non-synthetic fibers and polyolefins all throughout the bay across all sample types. So they're very dominant everywhere. For our morphologies, so fibers, fragments, films, etc., we realized that depending on where we are in the bay, the morphology proportions varied, indicating that perhaps depending on where we are in the bay, the importance of sources differs. So in Central and South Bay, we found huge amounts of fragments in surface water, but in contrast, in other regions, fibers dominated. We also conducted principal components analysis, PCA analysis, to explore how similar or how different the microplastic communities within our different samples were. For our sources PCA plot, we plotted all three water samples, so surface, stormwater, and wastewater together. We found that the overlap of the ellipses shows that surface water microplastic samples can be influenced by source water communities, so both wastewater and stormwater. So the overlap shows that within that region, those samples within the ellipses are very similar in microplastic community to each other. So the source waters do have some influence on what we're finding in what kinds of microplastics we're finding in surface water. In terms of our fate analyses, we plotted surface water, fish, and sediment samples on the same axes, and we found that characteristic microplastic morphologies tend to accumulate into characteristic parts of the bay. So specifically, lighter, floatier microplastic fragments are found in surface water. Black fragments, especially dense fragments, are found in sediment, whereas fibers tend to accumulate in fish. So our overall conclusions are that the bay contains high contamination levels of microplastics, which are influenced by environmental variables including seasonality, depth, and current velocity. We found that, strikingly, stormwater is a huge pathway of microplastics to the bay, but depending on where we are in the bay, both stormwater and wastewater can be important. We found that characteristic plastics are found in characteristic locations within the bay, and that Lower South Bay can be an overall important sink of all microplastics, and that to mitigate microplastics, we need specific strategies tailored to specific sources. And I just have some upstream sources listed here, solutions listed here, such as rain gardens, litter traps, and washing machine filters, but feel free to ask me about that later on. So I just want to conclude my talk by saying thank you very much to all the people here, and thank you for listening.